Good morning. As you may know from the previous videos, the IBL process consists of 5 phases: orientation, conceptualization, investigation, conclusion, and discussion. In this session, we will explore how to effectively implement it to foster deeper understanding and engagement in STEM learning. Going into each phase more in detail in terms of the type of activities to be carried out and the resources that can support the process. We will explore how to apply the IBL through a practical example adapted from an article published in the Biochemistry and Molecular Biology Education Journal. In this example, students of an undergraduate laboratory course were involved in an inquiry-based activity in which they explore a specific laboratory technique, reproducing firstly the experiment correctly, that is, following the standard protocol, and then repeating it by introducing procedural variation. For example, variation of the temperature or different dilution of sample that led to wrong results. The main goal is to promote a deeper understanding of scientific principles and experimental techniques through the analysis of the consequences of such modifications. The first phase of the IBL process, known as the orientation, is designed to promote students' interest and curiosity about a particular topic or phenomenon. For instance, in the context of this laboratory technique course, used as an example, students working in groups with the main objective to explore the key features of a specific laboratory technique assigned by the teacher. The goal of this phase is to help students develop a basic understanding of the laboratory technique, including the role of various factors as temperature, dilution, and others, throughout the different procedural steps. The following phase, the conceptualization, involves students in hypothesis formulation. In our example, students might be asked to hypothesize how the outcome of the standard procedure could change if variables such as the temperature and sample dilution are altered at specific stages. Formulating clear hypotheses is sometimes a challenge for university students who may lack a basic understanding of what an hypothesis is. To address this, teacher can provide structured guidance, including definitions and reflective indications, such as the teacher might pose the following question. A dependent variable is the factor that changes in response to independent variable. Reflecting on the laboratory procedure you are analyzing, what is the dependent variable? Alternatively, the teacher may decide to directly provide the hypothesis for students to test. The level and type of support should be tailored to the student's experience, skills, and prior knowledge. In the investigation phase, students move from curiosity to action, actively engaging in experiments to answer their hypothesis. This hands-on exploration may include observation, making predictions, conducting experiments, and interpreting results. Students develop and carry out a strategic plan to test their hypothesis, carefully manipulating and controlling specific variables. The phase concludes with data analysis, aimed at identifying the relationships among variables, helping students revisit their initial hypothesis and draw conclusions. In our example, students must establish what they should observe and which data need to be collected. They test both the standard procedures and the modified versions to assess how changes affect the final outcomes. The conclusion phase focuses on evaluating the results of the investigation. In this example, students compare the outcomes of the standards and modified laboratory procedures against the original research question or hypothesis formulated. Finally, during the discussion phase, Students are involved in an activity to reflect on the whole inquiry process or on a single step of the inquiry process and share their findings. This reflection helps students make sense of their learning and articulate what they discovered. Each group can present their conclusions regarding the laboratory procedures, and their reflection can be further supported by guiding questions as what did I do during this activity? What did I take of those actions? Was my process effective? What other strategies could I consider in a similar context? In conclusion, IBL is a valuable strategy for engaging students in authentic scientific practices and promoting learning in STEM. By understanding the different phases of the inquiry cycle and providing targeted support, teachers can effectively guide students to become competent and reflective. Thank you for your time.